Okay, so we continue with this sympatric speciation. So totally different from the allopatric speciation. Sympatric speciations occur when there is no physical barriers, okay? No physical barrier that preventing any member of the species from mating with the other. So it means that they are at a close proximity. They still can interbreed, okay? They still can interbreed, but they are within the close proximity. Okay, so you look at this. Okay, if you look at this, so originally they belongs to the same species, but due to certain particular events, they develop into two different species. Initially they actually belongs to the same species, one populations. Now from one populations, they becomes two different species. So the most common way we have to know here is through the polyploidy. Okay, so when I say polyploidy, polyploidy basically means that this organism, we have they have more than two complete set of chromosomes. So due to one of the events here called non-disjunction mutations, later I will show you guys, okay? And syllabus, we can term it as a 40 meiosis. So therefore, gametes end up with two sets of chromosomes rather than one set. So they form what we call the diploid gamete. So if these two, or with this diploid gamete fuse together, they're going to have the zygote. So this zygote will get four complete sets of the chromosome. Okay, so if four, then we call it tetraploid. Okay, so let us look at the whole process. Now, try to imagine what actually happened here is, so this is a male. Okay, male. So this is a female. So what will happen here is, you try to imagine, okay, so this is during the metaphase 1. So you can imagine that this is a cell. Okay, so with homologous chromosome, 2 blue, homologous chromosome, 2 red, homologous chromosome. Now we do know that after meiosis, okay, after meiosis, we should, meiosis 1, okay, after meiosis 1, so ignore the crossing over effects, we should get a haploid gamete, right? Segregations of the homologous chromosome take place. Cannot be segregated, right? Then after meiosis 2, then we get 4 cells. Now in this case, we actually ignore the effect of the crossing over and also the independent assortments. You can blue, okay, blue chromosome, blue chromosome, blue chromosome, and also the red chromosome. So this kind of gamete we call it as the haploid gamete. N. So this is the diploid cells, 2N. So the same thing will happen here. It will same thing will happen. So means that for female also produce, okay, the same thing, okay, for female. Let me rub it off here. So same thing, during the metaphase, they have 2 chromosome, okay, homologous blue chromosome, homologous red chromosome. So after meiosis 1, we get the haploid cells already. Okay, why haploids? Because we're going to contain 1 blue, 1 red. Okay, 1 blue, 1 red. Correct not? So if this continue the end of the meiosis 2, then we're going to get 4 haploid gametes. Okay, female gametes in this case. So blue color, blue color, blue color, and then red. Correct. Okay, so if the fertilization, this is a haploid gamete also. So if the fertilizations take place, between these two male and female gametes, we are going to get a zygote. So this zygote must be 2N because this zygote is going to get 1 blue, 2 blue, 1 red, 2 reds. Cannot. So 2 sets. Okay, clear? Okay, 2 sets. So zygote will be 2 ends, and then mitosis take place and development. We're going to develop into the multicellular organism. Okay, they're going to develop into this multicellular organism. Okay, so if you want to take a photo, you can. Okay, so now look at this. Male and female under normal conditions. Okay, later I'm going to show you how the polyploidy actually develop a polyploidy. Okay, so now next. So what do you mean by polyploidy? If you look at this, during the metaphase 1, again, metaphase 1. Same thing, we have 2 blue color chromosome, 2 red color. But because of the event called non-disjunction mutation, so means non-disjunction mutation, so by rights, we need to divide 1 chromosome, okay, another homologous chromosome, we have to divide, right? There's no divisions, okay? We don't divide them into 2 cells. What will happen? We move all of them into 1 side. So it means that at the ends of the meiosis 1, so in this case, you can see that non-disjunction mutation take place. Can I say non-disjunction mutation? So non-disjunction mutation take place, we're going to get this gamete now. Not gamete yet, but haploid cell, diploid cells here. So it means that this cell will have 2 blue instead of 1. Two red. All chromosome actually move to 1 side. Another one actually blank. No chromosome. Can I see that? So, and if let's say everything take place at the end, okay, at the ends of the meiosis 2, then you're going to get 2 cells or 2 gametes. These gametes now have 2 blue color chromosome and 2 red. So this kind of gametes is termed as the diploid gamete. 2 set, 1 blue, 1 red, 1 set, right? So this one we call it 2N gametes, or we call it a diploid gamete. Are you clear? It's called diploid gametes. So when these 2 diploid gametes fertilize together, always remember 1 blue plus 1 red, consider 1 set. Are you clear? So here we have diploid gametes. When fertilization takes place, let's say when male gamete, let's say this is a male gamete, this is a female gamete. So fertilization. If the fertilization takes place, what will happen? If the fertilization takes place, then you're going to get a tetraploid. You can see that. So there are 4 blue chromosome, 4 red chromosome. That's that. So in this plant or this zygote is 4N. It's called tetraploid. Okay, it's called tetraploid. So this tetraploid is sterile. Most of the times they're sterile because 4 chromosomes, they can't pair up during meiosis. They are not clever, 2 by 2 they pair up. So most of the time, I don't say 100%, sometimes they can, but most of the time they can't. Are you clear? So they are sterile, but if in the plant, no problem, they can still grow asexual reproduction. So it means that there are no problem to carry out mitosis so that they get into the multicellular multicellular organism or multicellular plant, no problem. Are you clear? So for plant, even better, because for plants, they can carry out asexual reproduction. So tetraploid, normally they can survive because mitosis does not involve. So question always asks here, why tetraploids sterile, but, okay, diploids, they are fertile? Because in this case, we always have to link it. When talk about sterile, any organism, when are sterile in this case, in this context, so we always link it with they can't carry out meiosis. Meiosis cannot, particularly when prophase 1. Why? Because no homologous chromosome can pair out. Now, the context here is very important. When we use the word pair out, means two. You must be chromosome like this. They can pair out. So if 4 of them, how to pair out? So very, very rare chance, very, very rare chance actually they know, oh, 2 pair up like this, another 2 pair up like this. Very, very rare. Because of protein-protein interactions, 4 of them will clump together, so that's why they can't produce. Okay? But occasionally, I say that very rare chance, if they can, if this multicellular organism, we do know that they are sterile. They can't produce. But in the case that they produce gametes, so this gamete again is a diploid gamete. Are you clear? They will still produce a diploid gamete. What I mean here is, look at this. So sometimes suddenly they are so clever. Blue chromosome. Okay, red chromosome. So it's very rare. It's very, very rare. Okay, so in this case, what will happen if you complete the meiosis? Again, they will produce diploid gametes again. Okay, they'll produce diploid gametes. So means that you're going to have 2 blue, 2 red. So this is a diploid gamete. Half makrana. Tetraploid means 4n. Tetraploid 4n. So this is diploid gamete. If a diploid gamete fertilized with a normal gamete, so what will happen here? Normal gametes, right? This is a haploid. And diploid 2n. So fertilization between these two will give us the tetraploid. Are you clear? Tetraploid. So it means that you're going to have 3 sets. 1, 3 blue, 3 red. So tetraploids confirm, 100% confirm they are sterile. If 4 cannot pair up, 3 how to pair? Right? 3 definitely you cannot pair. 4 you can say that, oh, occasionally can be 2-2, but tetraploids definitely sterile. They won't be able to pair up their chromosome. So 3 of them come together, so they can't produce gametes at all. Can I see that? So tetraploids. So now the idea here is nothing to do with speciation yet. No species developed because they are sterile. When they are sterile, basically they are not potentially interbred. So they are sterile yet. Okay, so they are non-species, new species. So what I want to say here is, because of the non-disjunction mutation here, can I see that? Because of the non-disjunction mutation that takes place here, it can produce what we call as the diploid gamete, 2n gamete. So when 2n and 2n together, we're going to get the tetraploid, which is more normally, we say that generally they are sterile. So they can continue with the mitosis, doesn't matter. But when they produce, in case they can produce, occasionally, again, they're going to produce diploid gametes. So they are diploid gametes. When the diploid gamete fertilized with the haploid gametes, we're going to get a tetraploid. So I'm going to introduce you guys a new term here. So just now we can see that original parental cells, parental cells originally 2n with 2 blue, 2 red. Can I see that? Blue and red. So if you look at the tetraploid just now, the first tetraploid that formed, Okay, sorry. Before this should be this better. Don't confuse you guys. So 2 blue. I mean no replication yet, like original. Okay, so tetraploid. 4 blue, 4 red. Okay, triploid. 3 blue, 3 red. Okay, you can see that. So this 4n, this is 3n. 4n and 3n. Can you, do you realize that they actually, all of them inherit the same set of chromosomes from one species? Original parental species only involve one parent, cannot. All blue and red, blue and red, blue and red. So this form of, okay, this form of the tetraploid, we name it as auto tetraploid. Auto here means that same from the one parent, same parent. Tetraploid means 4n. Okay, and auto tetraploid generally they are sterile. Be careful, because later you're going to get confused, because we have another one called allotetraploid. Allotetraploids, they are fertile. Now I just say auto tetraploid. Why? Because for them they can't pair up. Okay, generally, I don't say 100%, but generally they can't pair up during prophase 1. Therefore, they won't be able to produce gametes. So they are sterile or infertile. Same goes to these triploids. Same goes to these triploids. If you look at these triploids, they actually belong to the same species, original parental species, right? One parental species only, not from 2 parents. So it means that this triploid also named as autotriploid. So this autotriploid 3n triploid, okay, auto means on the same parent, one parent only, from one parent only. Again, this autotriploid, autotriploids definitely sterile 100%. Oops, okay, never mind. Okay, so they are sterile. Are you clear? 100% they are sterile. Okay, so at least you have a look here what I mean. So because from one same species, so we name it as auto. Okay, I highlight here auto tetraploids, auto tetraploid. Okay, so auto tetraploid and auto triploid. Basically, they are coming from the same species. And generally for tetraploid, we call it sterile because they can't produce gamete. Occasionally they can, but generally we say they are sterile. Okay, means that occasionally, so we still name it as sterile. Okay, so all auto triploids 100% sterile. Okay. So we go back to our notes here. So tetraploid form here this way are often sterile. Okay, not 100%, but often sterile. So as there are 4 of the same kind of chromosome, so they fail to pair up during prophase 1, so difficult for a cell to undergo meiosis and produce gametes, but they can grow perfectly because they can reproduce asexually. So it means that asexual reproductions only involve mitosis. So they involve mitosis. Mitosis does not require the homologous chromosome to pair up. So that's why mitosis can take place, but meiosis can't. Okay. So tetraploids. So occasionally, these tetraploid plants may manage to produce gametes. So these gametes will be diploid gametes. I just like say that. So if one of these diploid gametes fused with the haploid gamete, it's going to form a triploid 3n. So it may be able to grow normally, but it will certainly, in this case, 100% sterile. Just now, we say that generally they are sterile, but now this one 100% sterile. Well, three how to pair, they can't pair out. Okay, so draw a diagram to show how a triploid and tetraploid can be formed due to the faulty meiosis, or in this case, we call it non-disjunction mutation. So I've done this now, right? The drawing parts here. So if you want to draw, basically you can draw it like this. You can check back, I mean, my diagrams, okay? So you can draw it as this first, 2n. So with and 2 red. So because of the meiosis, okay, or non-disjunction mutation, okay, during the meiosis 1, so you get 2 cells. So because of non-disjunction mutations, you get 2 blue chromosome together and 2 red chromosome together. So, and then it's going to form a diploid gamete. So this one is termed as diploid, so 2n. So I say that it's a diploid gamete. So when these 2 diploid gametes fertilize together, let's say this is the male gamete, this is a female gamete. So if fertilization takes place, you're going to form tetraploid. One, two, three, 4. 2, 3, 4. Are you clear? So this one is termed as the tetraploid. And they get it from one parent only, right? Forget from one parent only, right? So these tetraploids, we also name it as auto tetraploid. Okay, auto tetraploids, so which is 4n. Okay, so auto tetraploids, we generally claim them they are sterile or infertile. But occasionally, if they form, occasionally these tetraploids don't know how, okay, just randomly, they can produce gamete if, I say that if completes meiosis. If they can complete the meiosis, then again, they are going to form, okay, they're going to form what we call the diploid gametes again. Okay, reduced by half, right? Okay, diploid gamete. So this is 2n. So just now this is 4n tetraploids. So get 2n gametes or diploid gametes again. So 2n. So if fertilizations with a haploid gamete, n. So what do we have? In this case, we are going to have a triploid, 3n. But because of same parents, right? Because some same parent, this triploid also known as autotriploid. And they are sterile. Okay, definitely they are sterile. Why sterile? Because they're going to have 3 chromosome, 3 set chromosome. So 3 set, how they can actually align. They can't align and they can't pair up during the prophase 1. So that's why confirm 100% sterile. Okay. So these kind of polyploids that are contain 4 set chromosome tetraploid or 3 sets, right? All from the same species, right? One same species, right? So it say auto polyploids, right? Auto polyploids or auto triploids or auto, okay, tetraploids. But polyploid can also be formed containing chromosomes from two or more closely related species known as polyploids. So meiosis can happen in all allotetraploids. So the chromosomes from each species are not quite identical. So therefore they can pair up, less muddled situations. So allopolyploid may be able to produce plenty of gametes. So we consider it is fertile. So one well-known instance of the speciation through allopolyploidy is a cord grass known as Spartina angelica and modern variety of wheat called Triticum aestivum. Okay, so we're going to look at this now. What it means by the Spartina angelica and how this Triticum aestivum actually in the wheats do have this allopolyploids. Okay, so we continue with this. Now, so in this class, we have this first one, okay? Now, whatever I explain here, I just will use a very simple way to explain, okay? So first, you can see that this is from species 1, the first species. Okay, species 1. So look at this. So let's say this is a male species 1. So 2n. 2n equal to 4. So this is a diploid. Please follow. This is a bit confused here. So 2 blue, 2 red. Okay, 2 blue, 2 red. So I have the species 2. So species 2, definitely their chromosome will be different. Okay, so that's why I don't use blue anymore. So in this case, I use green and orange. So 2 greens, 2 orange. So but they are closely related. 2 species, they are closely related, also diploid. So 2n equal to 4. Now, in this case, they've carried out meiosis, no problem. So they complete meiosis. So now you know that if they complete meiosis, they are going to form a haploid gametes. So they form haploid gamete. Normal meiosis, nothing happened, nothing special happened. So this is haploid gamete. So consists of 1 blue and 1 red. So same goes to species 2. Species 2 carry out meiosis as well. So they will get what we call the gametes. So 1 green, 1 orange. But okay, 1 set. So this one also haploid gamete N. Can you see that? Haploid gamete N, haploid gamete N. Now, if let's say pre-zygotic, because there are 2 different species, right? So it means that isolations have to take place. Reproductive isolation should take place. But pre-zygotic cannot. Means that they're temporal in terms of the timing for them to get fertile, same. The timing for that means the location, same. Behavioral, they can recognize each other. Okay, so gametes almost the same, mechanical, no problem. So fertilization actually take place. So if the fertilization takes place here, we call it hybridize. So what word we're going to use here is the inter. Inter means between 2 species. Interspecies hybridization take place. So when hybridize take place, you can see that now it form 2n or diploid zygote. You can see that they form diploid zygotes 2n. So these diploid zygotes, we're going to have blue chromosome, red chromosome, but they cannot pair up orange chromosome and also, orange is smaller one. Okay, green chromosome and orange. So in this case, we call this as the hybrid. What kind of hybrid? Your hybrid between 2 species, right? So it's a sterile hybrid. Why sterile? Because now you can see that blue and greens are no longer homologous. Blue chromosome and green, non-homologous. So they cannot pair up. Can you see that? They can't pair up. So therefore, non-homologous, they cannot pair up. So if they cannot pair up, means that they cannot produce gamete. So therefore, they are sterile. Okay, they are sterile. So this is a mechanism called as the hybrid sterility take place, correct? You don't want the hybrid to form a new species because these are two species. Let's say, for example, tiger and lion. Tiger and lion, two different species, and you form liger. So liger, they are sterile. Why they are sterile? Because sterile hybrid take place. Blue and green chromosome, they are non-homologous. Red and orange, they are non-homologous. Therefore, they won't be able to form. Okay, they form the zygotes, but the offspring is okay. They can no problems. Okay, mitosis and development, no problem. They can become a multicellular individual. Okay, they can become multicellular individual, but as I said that, they are still sterile. Sorry, what I'm doing here. Okay, they are still sterile. They won't be able to produce gamete at all. They won't be able to produce gamete. Okay. So now, what actually happened here is, if non-disjunction mutation take place. Now look at this. This individual. I'll try to imagine. Blue chromosome, red chromosome, green chromosome. They cannot pair up. And I purposely draw it so they are not pairing up. They can't pair up. But non-disjunction mutation take place. Move all chromosome to one side. Okay, so I say that non-disjunction mutation take place. If in case non-disjunction mutation take place. So what will happen here after meiosis 1? You're going to get blue, green, red, orange together. Blue, green is they are not pairing up. Red, orange together. They're not pairing up. They can't pair up. Okay, and then they complete the meiosis 2. When they complete meiosis 2, what will happen here is they're going to form diploid gametes again. So after non-disjunction mutation, we're going to get diploid gamete. 2N. Now, why is it called 2N? Now look at this. So one blue. Okay, I'll put it this way. One blue. Hey, where's my blue? One blue. One red. As one pair, right? Original blue and red, one pair. Green and orange, another pair. Can I see that? So this gamete is called diploid gametes 2N. Can you see that? This is called diploid gametes. One blue, one red, consider one pair. Blue plus red, one pair. Green plus orange, another pair. So it means that we get the diploid gametes. Why diploid? Because 2N, 2 set. Okay. Now, if this is a male gamete, this is a female gamete, and fertilization takes place. Now we're going to get 2 blue, which can pair now. 2 reds, which can, oh, red is a small color one. 2 reds, they can pair now. 2 greens, which they can pair, and 2 orange, they can pair. Can you see that? So how many pair? 1 blue, 1 green, blue and red. 1 blue and 1 red as one pair. Okay, another green and orange, consider another pair. So total we have 2 of them. Correct enough? So this is a tetraploid. Can I see that? It's 4 set. 4 set. So 4N equal to 8. So this is a tetraploid. Now this tetraploid, if you look at it again from the top, they are from 2 different species, blue and red, from species 1 green and orange from species 2. Therefore, these kinds of the tetraploids is termed as allo. Different allo means different parents. Allo tetraploid. And if you look at the structure of the chromosome, blue can pair with the blue, red can pair with the red, green can pair with green, orange can pair with orange. So allo tetraploid, they are fertile. So because they are fertile means that they are, when they are fertile, right? They are fertile, right? So when they are fertile means that they have potential to interbreed because they can form gametes, and also they are reproductively isolated from parental species. So means that this allo tetraploid now considered as a new species now. They consider as a new species. Are you clear? So when they consider as a new species, then they must be reproductively isolated from the parental species. They must be reproductively isolated from the parental species. Are you clear? So always remember this is the allo tetraploid. Allo tetraploid. So allo tetraploid is different from the auto. Auto, they are sterile, but allo, they are non-sterile. They are fertile because they can pair. They can produce gametes. Okay, now how they produce gametes? Blue and blue. Let me draw out. So with a segregation, get blue. Red and red, take one red. Green and greens, take one green. Orange and orange, take one orange. So again, it can form gamete. So meiosis can take place. A normal meiosis can take place. Okay, so we look at an example. One example that require you guys to know. So actually between two types of the grass. So one is the Spartina. Okay, Spartina maritima. Okay, Spartina maritima interbreeds. Okay, in natural, interbreed with Spartina. Okay, alterniflora. You need to remember this name, Spartina alterniflora. So how are we going to put it here? So this is a cell, so 2n. So I represent it as AA. Oh, sorry. Yeah. I represent it. So A is one set of chromosome, another A is another set. So diploid, so 2n. Okay, 2n. So they are fertile. Spartina and alterniflora, so I represent it with BB. Why BB? Different set, also 2n. So if meiosis takes place, they will produce a circle is a gamete. So this one is N, only one set, A chromosome, whole set. Okay, let's say for example, humans, 23. So 23 of them, I put it as A. So this one, meiosis takes place, get B. So this is N because it's a haploid gamete. N haploid. Okay, so if fertilization takes place, gamete, right? So fertilization, or we say hybridization here because two different species. So if hybridization takes place, you're going to get zygote AB. This is zygote. So these zygotes, if they develop, continue to form the mitosis, so they'll form the multicellular AB. But this multicellular AB, A and B cannot pair, right? A and B cannot pair up. So if you cannot pair up, so this one we call as the sterile hybrid. So this sterile hybrid, by right, shouldn't give them a species name, but according to our syllabus, this sterile hybrid is named as Spartina townsendii. But they can grow, right? They can carry out the mitosis, they can carry out the asexual reproductions and continue to grow, no problem. Until one day, this AB carry out non-disjunction mutation. It's a random process. Carry out non-disjunction mutations, so they produce a diploid gamete. By right, AB should be separated up, but now AB all together. So this is a diploid gamete. 2N. So if a diploid gamete fertilized with another diploid gamete, AB, fertilization, we're going to get AABB. So now can you see that AA and BB, they can pair up. Set A chromosome and set A chromosome can pair up. Set B and set B can pair up. So this one is 4N, tetraploid. And this tetraploid is more precisely because A from one species, B from another species. So it means that we are going to get allo tetraploid. Allo tetraploids, they are fertile. So a new species developed. So AABB, they can produce a gametes now because A between 2A, I can take 1A, between 2B, I can take B. So this one, look, so that's why we say that it can carry out meiosis now. As long as they can carry out meiosis and produce gametes, then they are considered a new species. And these tetraploids with the name Spartina angelica. Okay, so Spartina angelica. Okay, you can take a photo if you need here. Maybe a bit small, but okay. Yep. Okay, so start with start with the Spartina maritima and alterniflora. They're two different species closely related. Then they produce the haploid gametes, but this hybridization takes place, they form this sterile hybrids. But because of non-disjunction mutation or 40 meiosis, they can produce the diploid gametes. When two diploid gametes fertilize together, now they form the Spartina angelica, which is a tetraploid. This tetraploid is termed as the allo tetraploid, which is fertile. So it means that they can produce offspring now. Okay, they can produce offspring because during the meiosis, A and A pair up, B and B pair out. So therefore, they form the gametes. So these gametes can continue. So therefore, you can see that this Spartina angelica actually continue to survive. Okay. Now another example is the wheat. How we do? So wheat actually is basically because of humans. Humans actually is the one that cultivate it. So okay. So first, this one you do not need to know the scientific names. Okay. So Encon wheat. Encon wheat actually, okay, with the AA. Okay, I use again AA. So 2N equal to 14. Okay, Encon wheat actually fertilized with, okay, or crossed with a wild goat grass. Okay, wild goat grass type 1. Okay, so this one is BB. So 1B equal to 7. Okay, so 1A equal to 7 chromosome. So another A equal to 7. So 7 and 7, 14. B and B, so 2N equal to 14. So again, as I say that they can carry out the meiosis, no problem. Can carry out the meiosis, no problem. To produce a gamete A. This one to produce gamete B. So both gametes, they are haploid gametes. Haploid. So N equal to 7. N equal to 7. So now what happened here is hybridization take place. So this hybridization is going to produce AB. Now A and B, they cannot pair out, right? A and B cannot pair out, but they are sterile hybrid. Okay, we name it as sterile hybrids. So if we look at it, because A is 7 chromosome, B also 7 chromosome. So this one is 2N equal to 14, but they are sterile. Okay, they are sterile. But non-disjunction mutation actually take place, double up the chromosome. Okay, in the gametes, non-disjunction. If a non-disjunction take place, then they will produce diploid gametes. Circle, when a circle means gamete. So this is a diploid gamete. Okay, diploid gamete. So 2n equal to 14. So when these two diploid gametes fertilize together, okay, so you're going to form AABB. So this one now is 4n equal to 28. 4n equal to 28. So we call it as a tetraploid. Tetraploids, and more precisely is allo, because from two different parents, right? So allo tetraploid. And this, because A they can pair out, B can pair out, so they can carry out the meiosis to produce gametes AB. So means that these allotetraploids, they are fertile now because they can produce gametes. I mean, we are not talking about the productions of the gamete through non-disjunction mutation, but through the normal meiosis. Okay. Now, what will happen here is this AABB is named as ammoid. We give them the name called ammoid. What will happen here is ammoid actually we cross it again with another species of wild goat grass, but species number 2. Okay, species number 2, wild goat grass. So in this case, I name it as CC. So C equal to 7 chromosome, another C equal to 7 chromosome. So they can carry out meiosis, no problem, because they are diploid, 2n equal to 14. So carry out meiosis to produce gametes with C chromosome. So fertilization takes place now again. We purposely make it, okay? So in this case, hybridization takes place again. So these hybridizations allow us to produce A, B, C. So A, B, C. A is one set, B another set, C is another set. So 3n. 3n equal to 21. So again, it must be sterile because 3 set, how to, okay? So they are sterile hybrid. Okay. But if this ABC manage, because they manage to carry out non-disjunction mutation, again, because of non-disjunction mutation, they produce a triploid gametes now. Can you see that? ABC. So 3n equal to 21. So this one actually is a triploid gamete. So when these triploid gametes and another triploid gametes fertilize together, so fertilizations, they're going to produce AABBCC. AABBCC. So this one is 6n. 6n equal to 42. This one actually is called as a hexaploid. 6n hexaploid. Okay. Or more precisely, because AA from one species, BB from one species, CC from another species. So this hexaploid is the allo hexaploid. Okay, it's allo hexaploid. And this allo hexaploid, AA can pair up, BB can pair up, CC can pair up. So it means that they can carry out normal meiosis. You can carry out normal meiosis to get A, B, C. Are you clear? Okay, so that's why we call it as the fertile. Okay, allo hexaploid, they are fertile. AA pair up, we choose 1, BB pair up, we choose 1, CC pair up, we can choose 1 to form this diploid. Triploid gamete. So this AABBCC is now termed as a modern wheat. So how we produce the modern wheat now, which is a hexaploid with 6n equal to 42. Okay, so it will be too small if I do this. Perhaps I can half. You want to take a photo? So we have the inconvit, wild goat grass 1. Then we get the ammoid, which is the tetraploids crossed with the wild goat grass 2, and then we get the hexaploids, okay, which is the modern wheat or bread wheat. Okay, so come back to our notes here. So one well-documented instance of these speciations through the Polyploidy is a Spartina angelica, and the modern variety of wheat called Triticum estivum. So you can try, you can scroll. Okay. And interspecies hybridization leading to polyploidy of wheat A, B, and C. So just now I have done. So that's all for our class. Okay. So off the record.